Hi, everybody. This is the Prostate Cancer Lab, and we're honored to have David Hysong and Katherine or Kate Arline with us from Shepherd Bio or Shepherd Therapeutics. We always start now with a disclaimer. I'm going to actually not read the exact text. I'm going to go and basically say that this is the Chat GPT version. I ran it through Chat GPT, and it's all much more colloquial now. And it's basically, this is not medical advice. This discussion should not be considered as advice. You should consult your physician and your medical team for any decisions you would make. And the second part is that everything you can and will be made public. So everything that we say here will be published on our website. And so if you're concerned about that, don't speak and maybe turn off. If you're concerned about your image being shared, because we will share this recording, you could turn off your video. And with that, I will introduce David and Kate. I'm really excited by their presentation. They shared it with me in advance. I think it's very, very interesting. They are doing work in RNA Seq analysis, which identifies more biomarkers. Most of the patients we have in prostate cancer have a long journey, and they have hopefully many shots on goal. And the RNA analysis is a new area. DNA sequencing seems to become more commonplace, but RNA sequencing is still somewhat novel. And to the extent from a patient's point of view that it can identify additional biomarkers, that's all a good thing, because it may come up with novel treatments and better guidance on the treatments in hand. So I'm hoping that that will be a result. And with that, turn it over to David and Kate, who have some slides, and we'll introduce things, and then we'll go into Q&A.
Wonderful. Brett, thank you so much. Ladies and gentlemen, my name is David Hysong. I'm the founder and CEO at Shepherd. Like many of you, I began my journey as a patient. I was diagnosed with adenoid cystic carcinoma, which is a rare head and neck malignancy, about 8 years ago. And it was my experience as a patient in confronting the paucity of options, even at initial diagnosis for a rare cancer, let alone for metastatic refractory patients that I met along the journey that led to the creation of Shepherd. I partnered with a gentleman named Gene Williams, who was an early employee under Henry Tremere at Genzyme, the first rare disease-focused biotech. And we set out to build a, quote, Genzyme for rare cancers, but a oncology-focused development company focused on finding and creating the assets that would help patients that, from our standpoint, have been left out of the development paradigm, that did not have FDA-approved therapeutics, let alone NCCN treatment guidelines often. So we founded the company about 7 years ago, and this will be the story of our journey. I am joined by my right-hand Chief Strategy Officer and the architect of the Delve platform that we will be presenting about today, Kate Arline. I met Kate at the Harvard Innovation Lab, where she was running her own company, and her partner had recently been diagnosed with a rare brain cancer himself. It was that experience that led the two of us to meet, and that led us down this path together. So I'll ask Kate to introduce herself before I dive into the presentation.
Really a pleasure to meet all of you, and Brad, great to see you again. So we were so inspired by the hackathons that you ran that we were really, really excited to join here. So when I joined Shepherd, I didn't have a typical background. I had a background in technology and data science, and the questions that we're trying to answer were specifically around when you don't know the target of a cancer, when you don't have a lot of information around the biology, how do you begin to think about leveraging new or existing drugs? And so what that led us to build is a platform that integrates a lot of omic data, over 100,000 patient samples representing 161 cancers, and ask questions of that data in a different way that David will go through some of those mechanisms right now. But what we're able to come up with is, based specifically on the RNA, the answer to questions about will therapies that are near existing work, regardless of the presence or the absence of the canonical target of the drug. And that allows us to operate on a single patient basis, as well as for looking across multiple indications and saying, does this drug potentially have applicability in cancers in which it hasn't been studied?
Wonderful. So folks, I'm going to walk us through this presentation from a high level. Again, I did not come, Kate is the more technological scientific brain behind this. So we're going to tag team this as a team. Certainly many of the questions will be directed towards her, but I'll walk you through, I think we have about 7 or 8 slides that should tell the story relatively robustly, and then we can save plenty of time for Q&A if that makes sense. So let me pull up this presentation, Brad, and I should be Sharing the screen. Can everyone see this presentation? Wonderful. Thumbs up. All right. Great. So we'll dive into it. So again, the product of working across indications where you oftentimes had very little scientific funding, you didn't have known or validated targets, again, lack of therapeutic options and even treatment guidelines led us down the path that, again, Kate just outlined. So what we chose to become was to become a multi-omic data company focused on patient data first and foremost, and on doing this from both ends, the ability to understand the full RNA multi-omic impact of a specific therapeutic, and then also what the full quote RNA fingerprint of a patient was, and then how to match those two things together. So again, the approach is a multi-omic, it's a data science approach. It's an in silico approach validated by a wet lab that we have outside of the Boston area. All of that focused on bringing next generation precision medicine to patients where very few options exist. So our starting point.
What are the multi-omics? What are the omics and multi?
Yeah, primarily, like I said, we work on RNA. So that's going to be what really drives everything for us. But we also will take in whole exome, whole genome whenever possible and look for all of the known and variants of unknown significance and their potential impact on those therapy recommendations as well.
Thanks.
Sure. So again, our starting point from a rare oncology perspective, drug development perspective, was we oftentimes are dealing in indications without a validated target. On a personalized medicine standpoint, looking at the number and percentage of oncogenes that are the target of an FDA-approved therapy. The current CDS applications on the market, were wonderful if you did have a single point mutation, did have a target, something that was overexpressed where there was an approved inhibitor on the market. But for the vast majority of patients, we found that this oftentimes wasn't the case. That there either wasn't a therapy that a report was able to identify, or the patient then had progressed via a therapeutic journey or had a metastatic refractory journey such that a report was unable to identify any type of effective therapeutic. So again, the whole idea behind a single target-based approach to drug development, a single mutation-based or target-based approach to precision medicine was a starting point, and we saw a major inadequacy and an opportunity to take in a much more complex, robust set of data, approach this from a hypothesis, mutation, target agnostic standpoint, and really start to understand the full impact of a therapeutic, both its on-target effects, its off-target effects, additional kinases hit, et cetera. So the result of that is the platform, again, that we're presenting to you today, which is the Delve platform focused on RNA, as Kate mentioned. And so what Delve does, in a nutshell, trying to visualize this, is instead of a single target-based approach, we, across our library of assets, so we look at about 500 different assets for every single patient that we bring on-house. And we've taken the time to understand and to decode the full RNA impact of that therapeutic across all of those patient samples and indications that Kate mentioned. So from our seat, oftentimes an asset is looked at in a highly specific set of cancers, oftentimes ones that express a specific target of interest. And it oftentimes completely leaves out the full impact of that drug across other mutational profiles, other indications, other patients at various stages in that journey. So when we start looking at a patient or an asset, we do so from a completely agnostic approach, allowing the data to then tell us the full impact and then where therapeutics could be most effective. So a number of tools help us to do that. Delve automatically assesses the utility of a number of types of assets, FDA-approved drugs in oncology, certainly standards of care, known combination protocols, repurposed therapies, generics. We'll get into that when we show you what a sample report looks like. But basically, there's about 500 assets in the Delve repository to date. And so that then informs when we come up with a patient's recommendation. It is taking in, again, the RNA signature of impact of the drug with the patient's RNA fingerprint, matching those together, and oftentimes coming up with novel recommendations and novel combinations. As Kate mentioned, a big tenet of the company is the belief that RNA Seq is a much superior data source to DNA. That is it is capturing a much more robust, complex system of information, and that the ability to interrogate that information agnostically and taken in tandem. So not looking at a few hundred genes for a panel, but looking at more than 20,000 coding genes in tandem and understanding how all of those relationships interact with the therapeutic is really the key to the platform. So again, a number of convictions are listed here, but in terms of RNA versus DNA, we're really looking at the ability to determine mutational variants that are actually expressed in the tumor, identifying the gene expression and pathway signatures for individual tumors, as well as the tumor type in general, identifying gene fusions arising from chromosomal translocations, again, obtaining information of at least 20,000 protein coding genes faster and cheaper than other NGS processes, quantifying the genetic dysregulations with continuous and confidence interval-bound data, powering a wide variety of computational tools, including mathematical algorithms, machine learning, and decision-based models, and providing universal scaling compatibility as opposed to leveraging platforms such as microarray. This is a visualization of what the output around a specific therapeutic looks like. So when Delve is run on a therapeutic, we are looking at not just the canonical interactions, the known interactions and genes that are important of the therapeutic from literature and validated studies, but everything else that is happening. What we oftentimes is that GCVA is pulling out anywhere from. Was there a question? No, just background noise. I apologize for the pause. What we are often find is that GCVA is pulling out anywhere from 20 to 200 genes that taken in tandem and in combination are actually the full impact signature of that drug, are the actual biomarkers of that drug. So you can see that comparing the canonical interactions from CIVIC with the GCVA genes, you're looking at, again, a much more robust network, much more complex network. When you map this backwards and forwards, when you look at the GCVA-derived genes, we often find that they are sitting at nodes and hubs of up to several dozen literature-validated proteins that have been, again, validated in the literature and by a scientific experiment. When we run Delve for a specific patient, you can see everything that goes into the final output in the final Delve report. So again, GCVA is a first cutoff. And so we're running a therapy through asking, does the known RNA impact signature of this asset match and fulfill the criteria of the patient's RNA signature such that we believe this patient will have a higher than average response to the therapeutic in question. So a number of different decision science methods, a number of different analyses and expressions that are incorporated into every patient report, ultimately resulting in the recommendation that we'll walk through in just a moment, which is what a sample report looks like. This is the current output of a Delve patient report. So again, you can see that leveraging the patient's RNA signature, RNA sequencing data, we're looking at about at least 500 assets that we've analyzed for the patient themselves, looking at assets that only pass every single gate that we've mentioned previously, only pass the highest criteria for predicted efficacy. We look at all of the known oncology combinations that are out there from a physician adoption standpoint, so that we're trying to identify FDA-approved oncology assets, ideally with additional, oftentimes, clinical evidence or existing literature that can help support the recommendation, but ultimately coming up with what is the one best option for you and your cancer to come out of this. It is oftentimes a novel set of assets. It's oftentimes a novel combination. We're able to highlight, again, how the Delve identified targets interface with those, with the canonical known targets, pulling out, again, any additional clinical or literature-based evidence that can help make the decision alongside the physician. Kate, I'd love for you to pop in and provide a little bit more detail around this report.
Yeah, so the 500 drugs we analyzed to give some context to some of the questions that are in the chat, these include over 80 FDA-approved therapies, and these are pan-cancer. So it wouldn't just be drugs that are approved in prostate, it would be FDA approved for anything. And then we're really looking for, are those drugs that are approved in prostate or in other indications popping up for a specific patient? Or are there other drugs that really have been in clinical trials or haven't really been researched in the context of this cancer that look like they will impact the specific patient? And then answering the question, why? And out of all of that, and I should add, we also have repurposed drugs. So some of those, like you can think about metformin, mebendazole, some other things that have known anti-cancer activity. We have adjuvant drugs, which are really interesting in terms of they can be radiosensitizers, they can be anti-nausea meds, anti-seizure meds that for specific patients sometimes will come up as having the potential for anti-cancer activity as well. And we have done a lot of research around understanding which those drugs potentially have the highest impact in certain scenarios and the mechanisms by which that's happening. And then out of all of those drugs, we're also going to be looking to see what are the mechanisms of action that appear to be most important based on what passes the algorithm that David just described. So not did a drug pass and then nothing else in the same class passed, but did 4 out of 6 drugs in a specific class pass, or 5 out of 8, or is it just really 1 out of 5 and maybe that's not as important for this patient? And so that's one way that we ultimately come up with these identified drugs. It's not just what crosses the algorithm, but what really seems very, very important. And can we match that with different dysregulations at the omic level, either in the DNA or in the RNA? The known combinations of doctors obviously and very wisely want to work with drugs that have known interactions so that they know the dosing profile, they know the drugs are not going to be overly toxic together. And so we have 612 as of this moment combination protocols that have been used in the literature, used for various cancers, so that you can say these drugs are known to be safe and tolerable when used together in humans. It doesn't happen every time that one of those protocols is filled for a patient in terms of the drugs that pass the other gates in DELV. But when they do pass, that is something that is added onto the report. In this case, this patient unfortunately did not have any known combinations pass for her cancer. And so what we have here is a DELV identified combination of the drugs that passed all the other DELV gates, passed the algorithm, have significant mechanisms for her. And then we would want to discuss with the doctor the potential to use those drugs either sequentially or in combination in a safe and tolerable way to impact important things in her cancer. So on the left, you can see that we did not identify any FDA-approved therapies in her specific cancer, but we did identify a number of drugs that had a lot of interactions with her tumor and that had evidence in this specific cancer. So they're being studied in clinical trials. And we presented that evidence here in the additional information column. In the right, you'll see the string charts that were also on another slide showing that these drugs are connected with the dysregulations in her cancer at a really intricate level. And this chart, this report specifically doesn't have the p-values on it, but typically you have a very, very small p-value of the probability that you're going to get the interconnections that we identified by chance.
Could any other slides you'd like me to double back on, let you give any additional information before we start doing a question and answer?
No, I think this is great.
Brad, can we open up to the floor to start having a discussion?
I think we should start with, I thought you had a slide that would talk about access, because I know many of our patients will be saying, this all sounds great. How do I access your services?
We do. Final slide. So again, Shepherd built the platform out over a number of years. We treated our first patient via Mayo Clinic last October. So we've been treating patients we've been invited to join the V Childhood Cancer Consortium, which we've been a part of for the past year. I've been treating patients as part of their molecular tumor board. So about 40 different hospitals across the country that we help work with the physicians for refractory metastatic patients. The full rollout of DELV to the general public is coming sometime this summer. In the interim, prior to that, so if you go to our website right now, you'll see a landing page that says launching in May. Your cancer has a fingerprint, and we can help you decode it. Until then, anyone that is interested in leveraging a DELV report, we simply ask them to email us at delv@shepherd.bio. Again, right now we're operating off of third-party sequencing data. Becoming a one-stop shop CLIA certified lab is very much in the future, but right now we are operating on top of other third-party data, companies such as Tempus or someone similar. So basically, you can email us. We would work with you to have your third-party sequencing data sent to us, run the platform, and then generate the report.
That's Mike Yancey. Let me ask a question, because you mentioned Tempus, and I actually got a Tempus report, but then I asked for, I'll say the broader data spectrum, and of course via email, or not via email, but they gave me some links that were able to download a couple spreadsheets, which didn't look like a whole lot of information, but they also sent me information on a lot of large files that look like databases, which I'm assuming at this point probably have some of the data you need. So if I was able to work with you and give you access to that data and then confirm that it is what I think it is, you could do a report from that. Is that somewhat accurate?
Yep, that's right. So Tempus, we have great experience working with them, great company, and we're able to help you out talking with your account representative and get that data transferred to us. And then typically other sequencing providers are going to have the same type of process where you can request your data and then have it sent on to us. And like David said, ultimately this will be, coming all through Shepherd, but for right now, we're very happy to work with these vendors. And the other thing I want to mention is that the temporality of the sample is really important. So if you had a biopsy done when you were initially diagnosed, you went on to have a lot of different therapies, maybe you had some response to some and not some response to others, that's going to work less well for DELV than a biopsy that's very current, that is up to date. And you can imagine the reasons why. Obviously your tumor has changed in that time. And so we want to be looking at this pinpoint time at what's going on in your tumor and how the drugs are going to interact with all of those attributes right now. There's one caveat to that, which is if you've had a biopsy and you had some treatment after that biopsy, but you didn't have a significant response to that treatment, then in our experience with patients to date, that biopsy will still reflect enough of the tumor environment that delve can pull out good drug recommendations for you. But we're happy to talk with you individually about these questions as well.
I'd
Okay,
like to
thank
cue
you.
Brian.
Yeah, I've got,
I'd like
my
to.
biopsy.
I'm sorry. I wanted to cue Brian next, just to kind of lay out, since he knows the profiles of this dozen or so patients we have, how many of the 12, Brian, would be ready for a Shepherd kind of analysis where they have the detailed RNA Seq?
Just, off the top of my head, I would say probably half of them, ballpark. So 6 or so, maybe.
Yep.
Maybe a few more. I was also going to say, Mike, if you've got issues with Tempus, you shouldn't, but just loop me into that. Kate, I just had my whole exome sequenced through Tempus. And so I'm going to be following up with them this week to get that raw data. And so I'll work with you offline. There might be some synergies, Mike, with what you're doing too. So there might be a opportunity for a bundled request.
Okay, Brian, I'll get back in touch with you on that.
Awesome.
And Rick, yeah, go ahead, Rick. You've been trying to get your raw data to Shepherd as well. What's your perspective?
Oh, I dropped the ball. So I need to sign whatever I need to sign. I'm very excited about you guys, but yeah, that was my fault. So, but I wanted to raise my hand, and I plan to follow through. Hopefully we can do it together. It seems like you're offering this at no cost. Is that what I mean, it sounds like it's available, but Jason also asked, how much do we have to pay?
So when we launch and go live to the public, I think we're looking at a price point of somewhere between $2,500 and $3,500, depending on whether sequencing needs to be performed or not. The lower price point being if it's already ready and we simply are processing raw data. Prior to that, as part of this, we are definitely moving towards starting to charge patients. We've talked about patients that are incoming in the next few months, charging a flat $1,000 fee. That said, going forward, we will always make the commitment that if the patient is unable to pay, that we will still perform delve regardless. So that would be if someone was interested in having a delve report run right now, that would be, we'd look at $1,000 charge. But if that was a stopgap or a barrier for anyone, we would absolutely just ask you send us an email and we take care of it.
I think it'd be a barrier for just about everyone here. I'd have to think twice about it myself. Okay, so I did want to mention that Brian has 3 time points. So he's a unique guy that could be kind of, I think they were all done by Tempus.
Yep.
And so he's got primary, early metastases and a recent metastases. So
Correct.
it would be interesting to see the evolution. So just I thought I'd put that.
Yeah, I mean, thanks, Rick, for bringing that up. I mean, I know that my DNA has changed over that time period. I think with respect to the RNA that we analyzed between the first, between my primary and my first metastases, the genes that we looked at, there was the same gene expressions popped, but their expression relative to the pan-cancer cohort and the prostate cancer cohort changed.
Yeah.
But they were still,
Yeah. So I'll
relatively
answer
high.
this question first, and then Richard, your question in the chat as well, because I think they're a little bit connected. So we have definitely looked at patients with, I think, up to 4 different time points, and the expression, interestingly, can go up and down. You think of it kind of progressing in an upward fashion, at least with cell cycle and some of the things that are specific to the cancer. That doesn't always happen. So we definitely see changes. Sometimes the same panel of genes stays upregulated. Sometimes that shifts. We are always interested in that. So we'd always want to take a look at that if you have it, because there could be, if you have 4 time points and you had differential responses to treatment in between, there might be interesting information that can help guide, what was the cause for a lack of response or for the response. And on the DNA level, we have seen a patient I'm thinking of right now who had a fairly static cancer, a pediatric cancer, not a lot going on. His expression patterns changed, especially on the epigenetic side. And there was a mutation that developed in response to the monotherapy he had been on that we were able to go into the literature and say, this is probably the reason. And this is probably why you don't want to continue on this therapy. So it's always going to be individual for the cancer and the patient, but yes, we would definitely want to take a look at that. And then Richard, yes, we have dealt with patients who've had different biopsies coming from various parts of the body. That is always going to be preferable. So if you have multiple METs, getting them sequenced ideally from the same time point and having all of that data shared is extremely helpful. And what we do then is we run separate processes on each MET, and we look for the drugs that appear to have as much overlap as possible. So saying one drug is going to hit all three, and then there's one drug that's going to hit number 2 and 3, and another drug that's going to hit number 1 and 2, and trying to get as much coverage as possible and really, saturate those tumors with each of them with things that are going to be effective.
When you have that rare situation, rare because probably it's not going to happen in most patients. Do you find that typically there's a broad heterogeneity in the drug profiles, or do you find that most of the time the sort of drugs that work in one MET will work in another, or do you not have enough data to know?
I will say, A, I don't have enough data to know, and B, I've seen both things happen. And there seems to be a difference in cancers that are driven more specific. We've worked with a lot of PEDS patients, again, with the quieter tumors, and like David's cancer is a quieter tumor. And they tend to share more than a cancer that is coming from an environmental cause, specifically in adults, such as a metastatic lung cancer, because there's a lot more going on there, and you see frequently a lot more upregulation of more genes and many more mutations, obviously. So the answer can be all of the above.
And do cancers with a broader mutation profile, the well-known ones like melanoma, are those more likely to require, different drugs for different METs, or no rule like that?
No, not enough data, but it is more common in our experience so far to see that more complicated cancers require different drugs. And I'll say, depending on the specific cancer, we can work with multiple patients who've been pretreated with the same drugs and are at the same stage of disease. They've recurred or metastatic, and frequently the same drugs are coming up for different patients. So it could be 3 or 4 drugs, potentially some of them that have never really been studied in the cancer. And then for each patient, there are always also going to be some drugs that are only for that patient. And so that's really interesting from a clinical trial standpoint, because you can think about maybe a lot of patients are going to be helped by some drugs that need some more study. And also, we can add on some things that are very, very specific for the additional dysregulations in this cancer for this individual.
But you only have 500 drugs in a pharmacopoeia of several thousand, plus a bunch of other stuff that's probably useful, but not an approved drug, I guess. But then you said that some of these, I think you said, are non-oncology drugs. So it sounds like you are widening your panel to include, I don't know, metformin or whatever,
Yep.
Other stuff like that. Is your plan to eventually kind of cover the universe of drugs, but you got to start somewhere. So you start with the cancer drugs?
Yeah, we want to make sure that we have FDA-approved drugs that the patients can access. So we know everybody's not going to be eligible for every clinical trial. We know that there are lots of drugs out there that might not be available in different areas for different reasons or through different insurance. So we start with FDA approved. That being said, our library also includes clinical trial drugs, research tools, because again, we think the analysis of what appears to be important for a patient is significant in the ultimate outcome. And then we include some of the repurpose, like you're mentioning, and adjuvants. And yes, we do want to broaden that net. We've done a lot of research into repurposed therapies. We're big believers in specifically adding low toxicity therapies that have the potential for synergy and good patient outcomes, and just adding some of that power on top of the drugs that can be more toxic.
Kate, are you familiar with Reboot RX?
Yes, although I'm not familiar enough to speak cogently. So if you have a specific question, give me some
No,
context.
I just wanted to make sure that that connection was
Yes,
there.
yes.
I worked with Laura Kleiman
Oh,
to
yes, yes. You know
use
her well.
their
Yep, yep.
to use their technology to identify generic repurposable
Yes.
drugs in the
Yep.
prostate cancer setting. And so I know she's got a huge data set.
Yeah, I know. Yeah, she's done great work. I've been really excited to see all that progress. So we've met with them a couple times, and then the gentleman who runs the repurposing overall, anybody recall his name offhand? Because I met with him a few times as well. Anyway, huge fans. Yes, we love all of that.
Okay, cool. Yeah, I'm gonna get his name because it's gonna bother me.
Yep.
I know who you're talking about. Great guy.
I have a quick question on the test, and it's around, you can the output of this test is wonderful, but there's a few ways of how you can get there, and I would just love your view on the different approaches. And one approach is if you have live tissue, you can do functional testing on it and just test on a bunch of combinations, create a cell line theoretically, and then or this RNA, approach to kind of get to the same or similar output. So I'm trying to clarify, if you were one of us and had those two options, which one would you pick? I understand you're coming from the RNA side.
Yep.
And, are they complementary, or how should we think about these?
Well, I haven't been a patient, but I've been around a lot of patients, and I was a caregiver. And so I think it's pretty easy for me to step into your shoes and say, in all honesty, I would do everything. So I would do everything. And from our side, we have had, people say I've had an organoid generated from my tumor, and I will definitely look at that data if you're able to test drugs on that tumor. For us, because we look at the specific transcriptome coming out of the tumor from the bulk RNA, which, we all know that has strengths and weaknesses, but that transcriptome is going to be different from the generated organoid or cell line, because even if it's very close, even if the organoid is closer than, a typical cancer cell line, it's still going to have some changes. And so for DELV to predict, what we would like to do is have the sequencing from the organoid and the sequencing from the tumor, if we were to knit those together. But in your case, as a patient, I would say, look at what's in concert, look at what's in common between those two things and put more weight on that, because DELV would treat the organoid and the patient tumor as completely separate entities, and it might have different outputs on the organoid.
Yeah.
Okay. That makes sense.
Yeah, no, that does. And then on a competitive perspective, if you look at, your peers or other folks that are doing something like that, how do you think of yourself versus them, out of curiosity?
Yeah, so we're really focused on making sure every patient has therapies. And so David mentioned, there's a lot of great DNA testing that's gone on there. We know that those drugs can be really, really effective when you have specific mutations at giving you a great outcome. But there are also a lot of cancer patients who don't have those. And so we see ourselves as a supplement to those tests. So, obviously get that done as well, and then come to us and let's look at your RNA and let's talk to your doctor and figure out what is the best combination of those strategies. And then there are some other companies that are working on combinations and so forth. We overall just take a different approach to thinking about the data, and that, as David mentioned, we are agnostic to the tumor type and to the drug. And we're trying to connect all the things that are going on in the tumor, whether it's a typical driver, an atypical driver. And in the big picture here is that a drug can work in different ways for different patients. And so somebody getting a targeted therapy, you getting a targeted therapy, and somebody else in this room getting a targeted therapy are going to have different responses. And the goal of DELV is to identify how all the ways the drug works that have been studied and haven't been studied, and connect all of the things that are going on with your tumor in a network style of analysis, and come up with a higher probability test to answer the question, will you respond to this therapy that your DNA points to or a single marker points to in another context? So I think there's a big ideological difference in how this works. In practical standpoint, sometimes there is overlap between our recommendation or identified drugs and other companies' drugs, but we have never failed to match a patient with therapies, and that's a big distinguishing factor as well. So a Foundation Medicine style test, a Tempus test, especially with a quieter tumor, frequently there will be cases where you don't have a matched therapy, and that has not occurred for us to date.
There's a question in the chat that I'll take a gab at responding to, if I may. So Jita asked if there are any case studies with outcome data. So again, to date, a lot of our ability to look at the efficacy of a DELV recommendation, one is just how many physician adherence, whether they're leveraging the report or not, and their level of comfort with that. Two is whether the patient is receiving specifically Delve recommended drugs or that in addition to others. So on the Beach Childhood Cancer Consortium tumor boards we sit with, again, we are offering our recommendations alongside other analytic processes, other physician recommendations, and the physician, of course, is always ultimately responsible for choosing those. A couple of stories that we often tell the company. The first patient we treated was a refractory rhabdomyosarcoma patient, a young patient. She had ultimately been on 12 different therapeutics, 6 lines of therapy. All those had ultimately failed. She was in the ICU. She was intubated on oxygen. Her parents had been told to put her on hospice 3 times. We had her sequencing run, generated a report. The physician selected Delve drugs, a couple Delve drugs. 3 days later, she was out of the ICU. A few weeks later, she did fly home. Unfortunately, once home, her community physicians did change the protocol, took her off the Delve drugs, and so she ultimately did pass. So we were, again, very, very sorry to see that. We had a CNS patient that had a number of Mets around their spine. The physician ran a Delve report, selected 1 Delve recommended drug at a very low dose. 2 of those Mets fully necrosed. 2 of them remained stable for an entire year. And we are now joining that patient and physician to become a management partner. So we'll run a new Delve report, new sequencing, and then come up with new recommendations for the tumors that did begin to slowly grow. So the N of 1 case studies that we do have are relatively remarkable. But again, it's a small body that is continuing to grow to date. So.
And so, Rick, I see your question in the chat. I knew this was your question before I saw your name. So let me take these one by one. All right. So we start with the raw data. So we're going to get FastQ from Tempus or from anyone else you work with, because we want to run it on our pipeline and run everything consistently. So everything starts over from scratch. And then we do use, I'm going to say standard normalization methods, but I'm not going to go into them here. They're typical libraries, because the more important thing is that we have our internal normalization methods as well. And then we do a QC check to make sure that your sample is in line with what we need to make sure that there's nothing strange, there are no outliers. Yes, that has happened. So we put flags on it when it does happen. For immunotherapies, we are in the process to bring more immunotherapy abilities into Delve. So that's in process right now. We could give you the PD-1, PD-L1 markers if you're interested in that. We do not have a process to say, in the same way we do for small molecules, that we would anticipate a response or a lack of response. But I'll set that out there. That's going to be coming over the next, I'd say, 12 to 18 months. Transfact.
You should hang on. You should look into immune deconv in R.
Yes, we are working on deconvolution right now. So we're actually working with an immunotherapy company right now. So that's something we're bringing on board. We want to be very, very specific about the tools that we use, that we have rigorously tested in our lab and with retrospective data, and just be very deliberate about anything that we start using for patients and make sure it's up to our standards. So we have a toolbox for fun, more fun things, and we have a toolbox for the serious things, and we use the serious things on patients.
Before we go from normalization, what cohort do you use? So let's say you get counts or transcripts per million or whatever you get derived from your FastQ, and you say some number. It's either a count or a relative count. And that number is 100. So we've seen RNA Seq showing 100 instances. How do you know that's high or low?
Yeah, so that's a great question. And the short answer is I'm not going to be able to answer all of that right here, because it's a very long answer, and it comes to how is Delve different, from the previous question. And Delve is different in part because we're not comparing absolute counts, we're comparing distributions. So we're comparing distributions of the entire gene set and then distributions of individual genes, in contrast with a reference panel. And so the normalization that goes on is specific to the sequencing provider, specific to your particular sample. And the goal of that normalization is to bring it in line with our reference that we use to create and understand and interpret the distribution. And so that overcomes some of the issues of bringing in data from different sources.
I guess the crux of the question is, so we saw, I don't want to name the company, but they used a proprietary cohort to normalize. And then there's no way to validate what the great and powerful Oz did behind the curtain, because they used an internal cohort. And so it was like, okay, well, here's your answer. And it was very less than satisfying to say, okay, the great and powerful Oz told you this. And so I'm wondering, the nature of my question is getting at, could an outside entity cross-validate what you're doing and say, yeah, I get the same answer? Or
So
when they differ, how do we know what's right?
In the end part of your question, you're talking about using QIAGEN tools and other things. And so one important thing to mention here is that the really heavy duty tools are internal to Shepherd, that they were developed by us, validated by us, that we use the TCGA data. TCGA data specifically is our baseline normalization. And so when we have, so I put that out there, and that's in our poster and all, so you can go through that method. I think we probably shared it with you, and I'm happy to share that with any of you because it'll go a little more in the weeds on that. But we're also more sophisticated than that. And that's so there's some of the normalization methods we use are, again, because we need to normalize to a distribution, not to counts. But the baseline data and our pipeline itself is in reference to the Broad pipelines and data sets. Does that help? I'm happy to answer any more. I just want to make sure we get through everything.
Sure, yeah. And I know we're going into the weeds here, so
no, I'm happy to go through it.
In the interest of time and not to dominate, I'd say my last question right now is, what AI do you use?
So that's another really important part about Delve is we do not, as a standard practice, deploy any black box AI on patients. Our algorithms are white box. Means that we can go in and say why something is coming out. We can use the string charts you saw to relate it to what is being measured with standard bioinformatic processes as being dysregulated, either at the DNA or the genome level, or the transcriptome level. And we can dive into, does that map out with standard bioinformatic markers? Does that map out with the mutational analysis that is considered, in the literature? We also, on top of that algorithm that is white box and that is built on our normalization methods, have what we call drug to patient, which takes in 5,000 literature validated analytics that say this specific upregulation or downregulation or mutation is connected to response or resistance for a specific drug in a specific cancer. And we will add all that information in when we're making those final selections for the Delve identified therapies for an individual patient. So there are actually a lot of different tools that run. None of them are going to be your neural nets and your black box methods that would just give you kind of a metric to say yes or no. This is something that we can really open up, interrogate, and we're happy to share more of that with you as a patient. And then your question about string is actually important to that, because we operate at the transcriptome level, string operates at the protein level. And what string does, there can be things going on from a mutational impact that look important but aren't. And there can be dysregulations in the transcriptome that look important but aren't. But when we really distill it down to what does GCVA, the algorithm, pull out as being important about a drug, does that connect with what is known via standard traditional methods to be important with your cancer? When the answer is yes, via that type of analysis, then that is very impactful for our understanding.
So Kate, just to dovetail into that, you're looking at RNA. If we could integrate proteomic data into our profile and pair it with our RNA, or have you pair it with our RNA, how would that change your matching process?
It would become richer. So one reason we work on transcriptome is we consider it a richer data source than DNA for some of the reasons I listed, and it's a more complex data source right now. If we could get full protein analysis, or even if you have a limited protein panel, we would definitely integrate that into what we're doing for you.
Okay, so I'm just going to connect the dots here. So you heard from Mike Yancey, who's one of the patients here. He just worked with one of our partners, mProbe, that provides proteomic analysis, which he had done. They identified a drug that was previously off the radar for him. It was in a different setting, cancer setting, and he's currently on that drug right now. If he could get his RNA Seq data from Tempus, and I could potentially help him with that, I have my RNA Seq data. I'm going to have a proteomic report. I think what we can do is we might be able to bundle up some information for you that could be really powerful and maybe, push the boundaries of what you're doing.
Yeah, that would be fascinating. I'd love to do that. And Ricardo, that comes back to your question as well, which is what would you go with? And the answer is all of the above.
Yeah. Okay.
Is the output a CLIA certified report, or is it just like a report research for now?
So this is clinical decision support. So we are not operating a lab. So, we are working toward a CLIA certified lab where we would be handling the patient samples ourselves, but we don't do that right now. We just deal with data.
Okay, so you just provide like a report
Yep.
or something like that.
Yep.
Okay. And as a result, you don't need to take like a CLIA certified sequencing either.
Yes, we want CLIA certified sequencing.
So Ricardo, we're not providing that, but all the sequence data that we operate on has been performed by a CLIA certified lab.
Okay.
You touched on a couple of issues that we've dealt with on previous discussions. I just raise and see if you have any different points of view. One is drug combinations, which is a recurring theme here. And it's easy, it's relatively easy to identify them, and it's relatively difficult to get physicians to treat with them, to prescribe them.
Yep.
And the other is, you mentioned getting recent biopsies. And since prostate cancer patients, I think, what do we say, 6 or 8 out of 10 metastasize to the bones, getting bone biopsies is difficult often. Those are just 2 issues we've run into, the difficulty of getting current biopsy material for testing, and then the second, drug combinations, just in case you have any thoughts on either of those.
Yeah, I would say we echo that absolutely. And our prior call was also talking about the drug combinations. So that's, from the development standpoint, a lot of the work that we're doing is to identify known toxicity interactions and being able to eliminate any possibility of that preemptively from the report. And then I think the combinations in our experience are also down to the doctor's experience and interest in working with novel combinations versus not. And that's one reason we try to give more drugs than just here are your 4 drugs, or here's your one combination. We try to give enough latitude such that they're, given the status of the patient or things we don't know about the patient, that certain drugs might never be used, but maybe there's an alternative with the same mechanism that could be used. And so it's an ongoing, it's something that I don't know has one solution, but it's definitely at the top of our minds. And then the biopsy question is the same. And that's different for every cancer. So sometimes it's relatively easy to get a new biopsy, sometimes it's not. And the short answer is, every case is different, and we'll, do the best we can with whatever you're able to provide.
You mentioned 2 things that I thought I heard. I just wanted to clarify. One is, I think you said you don't work on biologics. And the other is sort of a question, well, I guess 3 questions. What does it take to bring a new drug into your system? Do you work on biologics? And you mentioned p-values, and without lab testing, I kind of wonder how you come up with p-values.
Okay, so I'll take the last one, the last one first, because it's the easiest. And the p-value is just the measure for the protein and GCV connections, the probability you'd see those connections by chance. So that's just the data science metric there. It's nothing that I'd say was clinically relevant.
It's
just
not a clinical p-value.
No, not at all.
It's just a network a systems biology link.
Yes, that's right. That's right. So if you have 3 connections and by chance you'd expect 1, then that's significant. But if it could be random, then we're not as interested in that drug.
And that's not, but there's no wet lab that tests that, except for you read the literature. You don't do the testing yourself.
So string isn't a fundamental part of DELV. It's an add-on for illustrative purposes. And string itself does have wet lab and literature validated connections as well as computational connections. So if you take a look at this deck, if Brad sends it out, you'll see that there are different colors on the lines relate to different levels of evidence. So a lot of that is actually lab validated.
Well, so I guess that gets into the question, what do you need to do to validate a new drug?
Yep. So that is also a question, similar to Rick's, about how GCVA works. And our inputs for new drugs are a big cell line panel, a high throughput screen panel of over 100 cell lines. And the idea here is, and then we're relating that to a response or resistance to the drug in vitro. And the reason that we use such a big panel is that it's, cell lines aren't great. We all know that, but they do capture real biological states. And the more of them you have, the more of those states that you are capturing. And what we're doing is comparing and contrasting between those cell lines, every possible interaction of every possible state of every cell line to come up with the solution set that is, when these things are really substantially upregulated and these other things are really substantially downregulated, various perturbations of those provide the solution set to the equation, what makes a drug work. And there's a lot of other validation work that goes on with that, but the basic input is cell line screening data and a big part of it, and then validation metrics alongside of it. So that's why we work predominantly with small molecules right now. There are some biologics that are in vitro accessible. The immunotherapies obviously are not. And so for immunotherapies or other things like that, what we are working toward is having clinical data, because when you have clinical data that says, here's the bulk transcriptome of the patient's biopsy prior to treatment, and here's whether or not that patient responded to Keytruda or whatever it is, then our algorithm can operate on that just the same way we operate on the other data and provide the equation that says this patient is likely to respond or not.
So you're not, so that's fascinating. You're not doing an in silico analysis to add a drug. You're actually doing the work with,
Yes.
with a cell line. Oh, wow. That's interesting. What does it cost to add a new drug, and how much time does it take?
It depends on the drug. So that's something where we have some great organizations that we work with, and depending, there's all kinds of data metrics here. We could have a separate panel on all of this, but some drugs are really easy to work with and some drugs are really hard. Some drugs, it's fine if we had 30 cell lines, and some drugs we need 250. And so we have some ways of projecting what that will be and which vendor we need to work with, and then making sure the data quality is there. So the cost can be anywhere between $20,000 and $250,000 to add a drug. And the timeline can be anywhere from a couple of weeks to several months.
Wow.
Richard, that's to generate that data de novo ourselves. Again, as Kate alluded to, the high level inputs are therapeutic response data with associated transcriptomic information, in vitro or patient data, or anywhere in between. So we've optimized for in vitro data because that was most readily accessible and what we were able to do for the majority of small molecules. Just as Kate mentioned, for biologics, immunotherapies that we're working on now and going forward, simply being able to pull in therapeutic response data with enough of a distribution of high responders, low responders, durable responders, non-durable responders, with the associated transcriptomic data for those patients, that would be enough to then see the platform, and actually would be an easier process than some of the additional work we have to do to then translate from the cell lines to then patient projections. But so if it's a small molecule that we need to run a screen on, exactly what Kate said, looking going forward, if we're able to work with other companies or pull data from the public domain as that becomes more readily available, that's simply what we need to access.
Yeah. So in a summary of that is, I would rather have human response data for every single drug and derive it from that than from cell line screens, but we'll get there when we get there. Because I think it's going to be more, or we can't say yet, but I think especially for certain classes of drugs, it'll be more reliable.
Well, human response data, but you'd have to have cell samples from those humans too, right?
Yep. So that's why we're partnering with other companies to do that for them as well.
We're almost out of time. Rick, you had a couple things in the chat. Do you want to ask the last question?
As long as no one else has a question, I've been too busy with my questions. And anyone else? Okay, what defines a connection?
So connection from the string charts, or connection for what are you responding to specifically there?
When I looked at your slide, I see green and blue and canonical interactions from Civic, but basically, and then I look at your final report and I see these circles, blue and green circles are connected with a line. Why are they there? Does this mean something passed a threshold and you saw from the data that there is a connection, or is that just a known string connection, or what's green and blue? And
Yeah, those are string charts. So I'm happy to walk you through those as well. Like I said, that's kind of an add-on. That's not how Delve operates. It's just something
Okay.
it makes it that's helpful to visualize it. And so what those are saying is there is
Okay.
a known biological net connection that's either been validated in the lab or proven in some other way between what GCVA pulled out for the drug and what is going on with the patient. And so at the patient level, the cutoff for connection would be the twofold change. And then you're coming up to the question again, what is your reference set? And for that, we will use whenever possible a matched set of tissue from the same sequencing provider or from the same cancer. There's a few different ways we cut that depending on how rare is the cancer, how many samples we have, or in the DNA level, it's going to be, this is a validated mutation. That has changed the expression level.
Well, I'm sold.
Okay, hey, great. I'm going to stop the recording, but I want to give Kate and David a chance for a final word, any kind of final message you want to leave us with.
Ms. Arline?
I'm really excited to work with y'all. Every patient we work with is a tremendous opportunity. You know, we do this because this is our mission in life, I believe, and we come to you as patients and caregivers and just helping to make things better and provide options where there haven't been good options before. So I'm excited to get this started and hopefully work with some of you individually.
We are too. Really impressive what you're doing, and the approach that you're taking is really it's awesome. And I think that you're building a great foundation. And I think it's just going to get better with more data sets that you bring into the equation.